Hi, so my name's Todd Stoltz. I have an odd job. I do business development and international relations for UC Davis Health. So now on to Jan. So Jan's a bit of a rock star when it comes to stem cells. She's been named as one of the top 50 global influencers in stem cells. We're very, very lucky to have her working at UC Davis Health. She is the director of the stem cell program for UC Davis School of Medicine. She is also the director of the Institute for Regenerative Cures there. She'll be talking lots more about what that means. And she's also the scientific director of the Good Manufacturing Practice facility at UC Davis. For those of you familiar with FDA regulations, good GMP manufacturing is a non-trivial endeavor, and there's a lot of work involved with keeping track of your process protocols. Because that's not quite enough to do, she is also the scientific director of the umbilical cord blood collection program for the entire state of California. Her research focuses on mesenchymal stem cells and applications of MSCs to various injury and disease. Some of her most interesting work is in Huntington's disease. You can find her talk from last year on the web about that if you want to explore that a little bit more. But today she'll be talking about some other topics. She is truly a translational researcher. She focuses on bench to bedside work, so she takes basic research discoveries and translates them into therapies that can be delivered at the bedside. As such, she is very, very much involved in a lot of clinical trials. So without further ado, I'll introduce Jan and take it from there.
Thank you, Todd. Appreciate it. Thank you, Todd. Well, I'm just delighted to be here again and to get to talk to you a little bit about what we're doing. And what I thought I would do is show you what we're doing in the stem cell field, and then kind of throw out the topics where we really need help with developing better technologies for growing the cells, administering the cells, monitoring the patients, and telemedicine, our favorite topic. So we are at the University of California Davis Medical Center. We're in Sacramento, actually, not in Davis. Have a lot of great facilities, and this one is my favorite, the UC Davis Institute for Regenerative Cures. Here we do a lot of team training. We have this Good Manufacturing Practice facility, as Todd had mentioned. It's a clean room facility for growing up the cells. And we also do a lot of research. So the overall goal of everything that we do is to improve therapies for our patients through teamwork and translational research. We work a lot with our UC Davis School of Veterinary Medicine. There are a lot of stem cell clinical trials ongoing for animals, and our School of Veterinary Medicine is really large, and so they can run a trial for instance, for German Shepherds with hip dysplasia, seeing how the stem cells might help with that. Cats with oral gingivitis, very severe, see how the stem cells can help with that. And they get enough patients that they can actually do controlled clinical trials, which is amazing. We can learn a lot from those large animals that are getting their own stem cells. So this is our program. If I seem a little scattered, I'm going to touch on just a few of these areas by the type of stem cell, and basically ask you for help using your incredibly technologically gifted minds, whereas we are biologists and work with cells, and maybe get your input on some things that we could do to better move the technology forward. So currently we have 24 stem cell or regenerative medicine clinical trials ongoing or recently completed, with over 20 in the pipeline. The green ones are the teams, the disease teams that have clinical trials, and the red are where we are actively talking to the Food and Drug Administration, at least with a pre-IND investigational new drug application in the works in the pipeline. So there's really a lot going on. And I had mentioned this GMP facility. Excuse me. And they do cellular product manufacturing for these clinical trials, trials that have US FDA approvals. That's a really important distinction, because you'll hear about these stem cell clinics that are popping up all over the United States that are using unregulated stem cells. They might take fat from someplace in the I point to myself here, fat from someplace in the patient's body and put it into their eye or someplace with absolutely no regulation. So everything that we do is with US FDA approved clinical trials. It's a very large GMP facility. We have 6 individual suites, and we work with a lot of academic and industry partners. We also have different cores if you're interested in doing this type of research and you need a core of specialized people to help you. I just wanted to mention that we do have these cores that run through university-approved recharge rates. We write a lot of letters for grants to help investigators move their research forward if they don't have, for instance, a humanized mouse facility at their institution. So starting with the types of stem cells, the blood-forming or hematopoietic stem cells are the stem cell that has been studied the longest. So these are the cells that are used in bone marrow transplantation, the first stem cell therapy that's been around for over 50 years now and is approved for insurance reimbursement. We have bone marrow stem cell gene therapy. This is where we take bone marrow or cord blood from a person or an infant. The cord blood would obviously come from a newborn. We insert a good copy of the broken gene, and if the child or person has a monogenic defect, one single gene that is faulty that's causing their disease, we can insert a vector, viral vector, with the gene of interest, a good copy of it. If it goes into the blood-forming stem cells, we can put it back into the patient's bloodstream. They home back into the bone marrow, and they'll make blood cell progeny for the lifetime of that patient, hopefully with the corrected gene expressing. So this is where I started my career 30 years ago. I hate to say it was that long ago, but working with Don Cohn we were at USC at that point. Don's now at UCLA. And we were working on bubble babies, and so using the cord blood from kids born with ADA deficiency, which is a terrible primary immune deficiency, inserting a copy of the good gene into their cord blood a couple days after they were born and then reinfusing it. I went on to have my career after 15 years with Don, excuse me, and he continued with this research and has now functionally cured over 30 children. So this is one of the examples of hematopoietic stem cell gene therapy and how it can work. So gene therapy has been through peaks and valleys over the 30 years that I've been in it, and we're now on a major upswing. So this is Biotech 2.0 for rare diseases this year. Of course, immunotherapies are really helping late-stage cancer patients have a second shot at survival. That's where the T cells from the patient are engineered to really aggressively attack the tumor. A lot of clinical trials, a lot of industry interest in that right now, in that type of gene therapy. One of the things that is really a bottleneck in the field is a virus shortage. So for any of these gene therapies or immunotherapies that you're doing, you have to have a viral vector carrying the copy of the gene of interest that you would transduce the cells. That means put the genes that the virus is carrying into the cell before readministration to the patients. And so there are a lot of people in the field working on engineering solutions to better and more effectively produce this virus. Sorry. What we're doing at UC Davis is the large-scale mesenchymal stem cell, which I'll talk about in a moment, and vector manufacturing using the Terumo quantum device. And this is an automated device. This is cell therapy 2.0, cell growing 2.0. So we used to have a lot of stacks of flasks, and now we have this one machine. It's a hollow fiber bioreactor. The cells that make the viral vector are growing on many, many fibers in 3 dimension, and will put out the virus, the live virus, into the media that is then collected. We can concentrate it and use it for the gene therapy clinical trials, and we've published on that. So this is really interesting because this is how to do cell growth if you don't want to do a lot of flasks, like a room full of stacks of flasks or cell hotels. The media hangs in one bag, the hollow fiber bioreactor is inside, the chamber is inside, with some saline, and then the waste comes out here, and when it needs either of these bags changed, it calls you on your cell phone. So that's technology 2.0 for growing cells rather than having pipettes and spending all day at the hood. A little more expensive right now, but in our GMP facility, well worth it. So these are the teams that are using hematopoietic stem cell therapy at UC Davis, and we have several of them in clinical trials. Now, the pluripotent cells are the ones that grab all the headlines. These are the embryonic stem cells that have been under debate for a long time. This is why the California Institute for Regenerative Medicine, that funds much of our research, was initially funded here in California, putting us really at the forefront of that research. So embryonic stem cells and induced pluripotent stem cells, and those are cells, of course, that you can take from the patient's skin, a little skin sample, grow out the dermal fibroblasts, add some genes to revert them to a more primitive state, and then you have a cell line that grows much like embryonic stem cells, and you can differentiate them down any type of lineage by adding growth factors at the right hour. A great job for grad students, adding growth factors at the right hour in their development. You can make a whole plate of neurons or a whole plate of hepatocytes, for instance. So we have a project ongoing for patients with liver fibrosis. We hope to get to patients in the future. This is a really nasty disease. The liver gets very cirrhotic and can't heal itself after a while. It's, you know, the cells that could regenerate themselves are like the seeds, but once the soil is also destroyed in the liver, it's just cannot regenerate anymore. So transplantation is the only option, but we don't have enough organs for transplantation for everyone on the waiting list. So we've been working on an IPSC-derived liver organoid technology. So we start out with skin fibroblasts from the patient, make these induced pluripotent stem cells by adding some genes here, grow out patient-specific human hepatocytes in culture, put them on some type of matrix or scaffold and transplant into the patient. And right now our patients are immune deficient mice that cannot reject these human organoids. And this is the work of Ping Zhu in our group. She's worked with me for a decade now on embryonic and iPSC technology. And so she's making these liver organoids, and these are from iPSC-derived hepatocytes. You mix them with iPSC-derived endothelial cells, which will make the vasculature. And so you get these little blobs of tissue forming. These are very small. They're like the size of my pinky nail. And in order to make a lot of them for a human patient, we'll need new technology developing stirred tank bioreactors with the appropriate glucose and minerals and proteins that these things need, and with oxygen to be developing. So people are working on that. We don't have very effective ones for this quite yet, but that's a technology that we need in the future. Right now, these are just grown in culture with changing the media manually every few hours. Again, great for graduate students. Once they're transplanted into immune deficient mice that have damaged liver, they can grow healthy, vascularized little nodules of human liver tissue. And the vascularization is the key thing and the key advance that Ping has developed, because without the blood supply going through the organ, it really limits the size that it can get to. So we're now working on scaling this up to have networks of these things in a larger animal. And you can see in this slide the beautiful vasculature that is growing through these organoids. So again, we need better technology to grow a lot of these in a big bioreactor, and we might be able to seed a matrix that could function as a human liver, or transplant them into the damaged liver. And so we do have several groups that are working toward iPSC or embryonic stem cell therapies to take them toward the clinic. We don't have any of these pluripotent stem cell trials in the clinic quite yet, but working on it. So the mesenchymal stem cells, as Todd had mentioned, are probably my favorite cell type, and that's because they are in a lot of clinical trials. They're very versatile, and I'll tell you what a few of our teams are doing with them. So they do have, these are the broad flat cells that line the bone marrow and make growth factors that support the hematopoietic stem cells. They also have these paramedic functions in helping damaged tissues heal, and especially when the damaged tissue does not have enough oxygen. So we get them from a normal donor bone marrow. We can grow up a big batch, in that Terumo bioreactor or in cell hotels, which are big stacks of flasks. They know how to home in on sick and dying cells. They are somewhat immunologically privileged. So they actively shelter themselves from the host immune system after transplant, which means that they can be transplanted. We can make a big batch from a normal healthy donor, qualify it, and use it for many patients. They are very good at regulating inflammation. They secrete factors that help nerves regenerate and to revascularize. I need some for my lungs, I think, today.